Good morning and salam sejahtera to Dr. Effendi and my fellow friends. Today, I will present my mock proposal entitled Phytochemical Screening and Preliminary Antioxidant Activity of Melastoma malabatricum leaf. Now, we're moving to the introduction. Melastoma malabatricum, commonly known as indudo, is a medicinal plant widely found in Malaysia. Traditionally, this plant has been used to treat wounds, diarrhea, and inflammation. Previous studies reported that Melastoma malabatricum contains important phytochemical compounds such as phenolic, tannins, and flavonoids. These compounds are known to contribute to antioxidant activity and help plant against environmental stress. And due to these beneficial properties, Melastoma malabatricum has gained increasing research interest. However, there are still some limitations in previous studies. Although many studies have investigated the phytochemical screening and antioxidant activity of Melastoma malabatricum, most only focuses on a general phytochemical screening or a single leaf stage. Comparative information regarding how leaf maturity influences the phytochemical composition and antioxidant activity are still limited. Therefore, there is a need to compare phytochemical composition and antioxidant activity among young, mature, and old leaves of Melastoma malabatricum. Based on this issue, three objectives were developed. The first objective is to identify the phytochemical compounds present in the leaf of Melastoma malabatricum. Second is to compare the phytochemical compounds of young, mature, and old leaves. Lastly, the third objective is to evaluate and compare the antioxidant activity of leaf extract at different stages. Next, I will briefly discuss findings from previous study. Previous studies reported that Melastoma malabatricum contains phytochemical compounds such as flavonoids, phenolics, and tannins, which contribute to antioxidant activity. In addition, antioxidant activity of this plant is commonly evaluated using assays such as DPPH, FRAP, and ABTS. Among these, DPPH assay is commonly used to evaluate free radical scavenging activity. Furthermore, studies in other plant species found that younger leaves may contain higher levels of defensive metabolites because they are more vulnerable to environmental stress and herbivore. However, comparative studies involving different leaf stages of Melastoma malabatricum remain limited. And due to these beneficial properties, Melastoma malabatricum has gained increasing research interest. However, there are still some limitations in previous studies. Now, I will explain the methodology used in this study. I've divided the methodology into 4 parts. The first part is sample collection and extraction. Fresh leaf of Melastoma malabatricum will be collected from Unimas arboretum. The leaf then will be categorized into 3 stages, which is young, mature, and old leaf, based on its morphological characteristic. After collection, the sample will be washed, air-dried, and ground into fine powder. The extraction process will then be carried out using 95% ethanol through the maceration method for 48 hours to obtain crude extracts. Now we're moving to the second part, which is phytochemical screening. Phytochemical screening will be performed to identify selected phytochemical compounds present in the leaf extract. Flavonoid will be tested using sodium hydroxide, phenolic will be tested using ferric chloride, and saponin will be determined using foam test. The presence or absence of phytochemical compounds will be recorded qualitatively based on color changes or foam formation. Moving to the next part, which is antioxidant activity. The antioxidant activity of leaf extract will be determined using DPPH radical scavenging assay. In this method, a methanol-based DPPH solution will be mixed with leaf extract and incubated in dark condition for 30 minutes. After the incubation, absorbance reading will be measured at 517 nanometer using a spectrophotometer. Then, the DPPH inhibition percentage will be calculated to determine the antioxidant activity. And finally, the last part of this method. The collected data will be analyzed statistically. All experiments will be conducted in triplicate and result will be expressed as mean with standard deviation. One-way ANOVA will be used to compare antioxidant activity among young, mature, and olive stages with significant levels set at P less than 0.05. Based on this methodology, several outcomes are expected. First, the study is expected to show the presence of phytochemical compounds, especially flavonoids and phenolic, in Melastoma malabatricum leaves. In addition, significant differences among leaf stages are also expected, with younger leaves potentially showing stronger antioxidant activity. Lastly, this slide shows proposed work schedule for this study, beginning with proposal preparation, followed by sample collection, laboratory analysis, data analysis, and report writing. And in conclusion, this study may provide better understanding regarding the effect of leaf maturity on phytochemical composition and antioxidant activity in Melastoma malabatricum. That's all from me. Thank you.